Hello, this is Chapter 8, Microbiology, Microbial Metabolism, Part 1. So, metabolism means the entirety of the chemical reactions going on inside a cell. And the chemical reactions can be split into two kinds. One is called catabolism, which means breaking down. So, we have some nutrient particles coming into the cell, we break them down into smaller molecules. So they can be used for energy or they can be used for building things, different things that the cell needs. But any catabolism, we are going to release stored energy. That's the thing about breaking molecules down, is that we can harness energy in those big molecules. And when we harness that energy, what the cell does with a lot of that energy is to make ATP. So ATP is a hugely important molecule. It is the energy currency of the cell. It's kind of like a charged battery. ATP stands for adenosine triphosphate. So it has three phosphate groups. As you can see, it's kind of like three bumps here. And those three phosphate groups are very energetic. So it turns out that the cell over evolution has basically learned or evolved to use ATP as its primary source of energy to power the other kind of reactions in the cell, which is anabolism, which is building up. So we're taking smaller precursor molecules, we're building the things that the cell needs and requires. So we have larger building blocks to make our macromolecules, which is something like protein or lipids, large carbohydrates, whatever the cell needs. So catabolism, in short, is breaking down, releasing stored energy. That stored energy is going to be used to make ATP, our energy currency of the cell, right? The ATP is going to power any chemical reaction that needs energy inside the cell. So ATP is paramountly important. So any anabolic activity that needs energy comes from ATP. When ATP is used, it basically just cleaves a phosphate group off of the ATP. So we have three phosphate groups in triphosphate. When we cleave one off, it's adenosine diphosphate. It has two phosphate groups. So all we need to do to recharge this ADP is to put a phosphate group back on. So that takes energy. It takes a lot of energy, and the energy comes from catabolism because we release that stored energy. That stored energy is used to put that phosphate group back onto ATP, so we have a charged battery. So we can kind of think of it as like a charged energetic molecule and sort of a depleted energy molecule. In terms of nutrition sources, there are these categories you want to know. So the mode of nutrition, there are autotrophs and heterotrophs. Autotrophs are self-feeders, and heterotrophs are other feeders. So in short, autotrophs can make their own carbon-based molecules. When you think about carbon-based molecules, right, things like proteins, sugars, fats, those all have a backbone of carbon, so they're organic. And if you're a self-feeder, you can create your own organic molecules using an inorganic precursor. So carbon dioxide here, and both of our autotrophs that can self-feed can use carbon dioxide. And, the key word is that it's an inorganic source, inorganic. Just think about how plants, they just need carbon dioxide in the air, the sunlight, and water, and then they can actually produce glucose, a sugar that they can use to break down for energy or to grow. So that's our autotroph part. The photo and the chemo refers to how do they get their energy, right? How do they get their energy to make that carbon? Well, light is for photo, right? So this is, if you think about plants, they are photoautotrophs. They take sunlight, they harness that sunlight so they can power the production of their organic molecules, of their carbon, using the inorganic precursor molecule. And then there are cells out there that are chemoautotrophs, which is kind of interesting. They can actually break down inorganic compounds, so kind of like salts and minerals and and things in the soil and rocks, they can actually break those minerals down as well. So that's their energy source. And then they use that to make their organic molecules using CO2. So these are going to be certain bacteria, prokaryotes. So we don't run across these very much in everyday life. So that's why it sounds kind of weird. The second category are heterotrophs, are other feeders. So we see that there are organic compounds, like what we use to build our bodies, has to be gotten by taking it in from the outside. So basically through our food. So that's why heterotroph is used here. Hetero is other feeder. So we need to eat other things in order to get our organic compounds. So we can't rely on carbon dioxide as a gas in the air. We can't take that in and then make sugar, right? That's impossible. But plants can. The energy to do this, right? So let's start with the obvious one. Chemoheterotrophs, such as animals and fungi and protists, they have to take in or basically eat their organic compounds to make other organic compounds. So breaking down organic compounds like food, that gives us energy, and the energy is used to make organic compounds we need for the cell. Other organisms can use light, and these are again some weird bacteria out there, but they can actually harness light as energy and use that energy to make organic compounds. But they also have to use organic compounds to take apart some organic compounds, put them together to make other organic compounds. So they still need to digest organic substances from the outside to make a base for their molecules, but they can use light as their energy source. So you should know what each of these mean. You don't need to memorize the specific types of organisms here, but you should know that what plants are. You should know that protists and fungi are going to be chemoheterotrophs. All right, so we're looking at ATP. This is just a simple cycle of ATP. So we take our energy that we consume either as sun or as food. When we break that down, we can add a phosphate group to ADP. So let's look down here, right? We have the ADP, that diphosphate with two phosphates. All we want to do is put that phosphate back on so that we can power up our charged molecule, the ATP. Putting a phosphate on a molecule is called phosphorylation. When the cell requires energy, it takes that molecule ATP and it basically just breaks off that last phosphate group. That breakage allows energy to be available for anabolism, for any kind of cellular work, for chemical reactions to happen. So when that's cleaved off, that's called dephosphorylation. All right, so it's just a cycle. So we have ATP, it gets used for energy. We took off a phosphate group, dephosphorylated. We have ADP, we need to power it back up. So we take the energy from our food to phosphorylate our ATP. The second idea or big subject in this unit is enzymes. So enzymes are biological catalysts. A catalyst, right, like in chemistry, is anything that makes a reaction go faster. And for specifically enzymes, they lower something called the activation energy. Enzymes are proteins, therefore they are sensitive to their environment and they can be altered or denatured by their environment. These are the three major things that can denature enzymes: that a rise in temperature, so heat, a change in pH, and a change in salt concentration. They are substrate specific. So one enzyme can only catalyze one chemical reaction. They're very specific to one very particular chemical reaction. So if you think about how many chemical reactions must be happening in our cells, there are so many enzymes that we are constantly making to make those chemical reactions happen. Enzymes are not used up in chemical reactions, so they can be reused over and over again. So enzymes are just a way for the reaction to proceed faster, and they're not destroyed at all. They're often named after their substrate and end in ASE. For example, Lactose, the sugar that's found in dairy and milk, is broken down by lactase. So you see how the name is really similar to the substrate and also ends in ASE. Not every single time, but most enzymes will have that naming procedure. And let's see. Oh yeah, one word I was using was substrate. The word substrate is just a term used for the reactants in a chemical reaction. So when we talk about enzymes, we don't say the reactants bind to them, we say the substrates bind to them. So we'll look at that. This is that reaction energy. So this is a graph of this idea that they're lowering the activation energy. So I have an example here. So glucose and fructose are both monomers. So they're just little individual sugar molecules. And then we want to put them together to make sucrose. This is table sugar. So when they combine chemically, we have a different sugar called sucrose. And in order to do the reaction, it actually takes a lot of energy input to get glucose and fructose close together, to face them the right way, to destabilize their bonds, to get them to react. And all of that is called activation energy. And so you can see that in this graph, we have a green line that represents without a catalyst or without an enzyme. And we also have the graph with a red line with an enzyme or with a catalyst. So notice how big the energy, activation energy is without the enzyme. It takes a lot of energy to do that. But with an enzyme, okay, the activation energy is very low. It's, well, not very low, but it's much lower, right? You can see the energy hump is depressed or lowered. So this is what enzymes do. Enzymes take that big hump of activation energy that is usually quite large, and it just pushes it way down. So not that much energy is required to make the reaction happen. So the reactions happen quicker and faster. And, all of life, the cells need reactions to happen almost immediately at times, right? So we need those enzymes to help reactions go faster. In terms of how enzymes work, enzymes have a protein base to them. So this is our protein, okay? The protein has a special area called the active site where the substrates will bind. So remember, the substrates is the same thing as the reactants. Okay, so just the building blocks, reactants. All right. And the substrates will come in and they'll fit the active site so that they are attracted to the active site and they'll bind there. But upon binding, the enzyme kind of hugs them a little bit closer. The enzyme changes shape slightly as the substrate binds, basically hugging them in, keeping them in. And that is called an induced fit model. So it's not a perfect fit here, but it's somewhat good enough to get the substrate in, and then they're really cradled and held together. And then when they are in the active site, that's where the chemical reaction happens. So there is the substrate is converted to the products. And so you see the color change. These are the products. And when the products are produced, they don't fit the active site very well anymore. So they're going to leave and then offer the enzyme an empty active site so it can be reused if another substrate is around. Okay. Some